So in this video, what we're going to be covering is our transfer technique. So to get started, you're going to do what you should always do when you come into lab and disinfect your work area. So just taking some diluted bleach and now wiping down the area which I'm going to be working in. So in this video, in aseptic technique, what we're going to start doing now when working with our bacteria is working in a flamed environment. So I'm going to turn on my gas. With my flint, I'm going to flame. So when working with a flame, you want to make sure not to get too close to it, is to burn yourself. You also don't want to put your sample in the flame unless you mean to, because this can cause some samples to lice or blow up in micro cases. And it can also create what's known as an aerosol. So what this is it's a particulate expansion of what's in your dish into the air. So that way you can actually infect yourself because, like we just said, aerosols are airborne. So then you could actually breathe in what you're working with. So you want to try to avoid that. So when working with a flame, there's an effect that occurs. So if I have a sample, and I have it right here, my sample is in a sterile environment. However, if I have my sample out here, it's starting to get a little bit past my sterile environment, because what happens is as this flame occurs, there's a phenomenon where it uplifts air, so air comes up and then it comes down outside a range of the flame itself. So that's why you always want to work inside the flame as best you can. So when you're inoculating your bacteria, you're going to need to have a sterile loop. So here with the metal loops that we use in the lab, to sterilize these, what we do is we flame them. So as this loop is turning an orange color in the flame, it's burning off and killing any microbes that might be remaining. So now, once I've sterilized this, I need to wait about 30 seconds to a minute before I can actually use this loop, because it will be too hot and it will kill the bacteria that we're trying to collect. So now what we're going to do, since we've waited, is we're going to do a broth-to-broth inoculation. So here I have my Serratia and I have LB media, okay? So to open this and to collect, what I'm going to do is I'm going to take one hand and remove the cap of the tube, and what I'm going to do is flame the top, okay? Now what I'm going to do is I'm going to take my sterile loop, I'm going to push it into the liquid media just to get a small amount, and then flame my tube before I close it. And then, again, get my sterile media, flame the top, take my inoculating loop, put it into my media, and just give it a good flick. I'm then going to flame the top of my tube, close it, and now what I'm going to do is label my tube. And now we'll use a permanent marker to label what we just inoculated, and then the date and our initials. So now that we've labeled our tube, we'll place it back onto our rack and we'll incubate it for growth. So now what we're going to do is we're going to inoculate an agar slant. So an agar slant is in a tube, and as you can see, it's slanted, hence the name agar slant. So again, we're going to take our bacteria, we're going to collect it on our loop, we're going to flame, close. And here, we're going to flame again. Now what we do is get our inoculating loop, put it onto our agar, and just swipe it across. It's very important to be sure not to stab your inoculating loop into the agar during this inoculation. And then once I have flamed the cap as I did, I would label my sample, have this inoculation incubated at 37 degrees Celsius. So here I have a pre-inoculated LB slant, Micrococcus luteus. So as you can see, this slant has bacterial growth on the surface of it. So we'll be using this slant now to inoculate a broth. So a slant to broth inoculation of Micrococcus luteus. So what I'll do is I take my sterile loop, I simply go in and I just need a small amount, so I lightly touch it. I lightly touch my grown-up Micrococcus luteus from the slant. I flame it, and then I take my sterile LV media that I have, flame the opening, and then again, get my loop, and I flick it in, I flame, and then I close, and then again, I label. So now what I'm going to do is I'm going to inoculate slant to slant. So I'm going to take my same Micrococcus luteus slant that I used previously to inoculate a broth. I have my sterile loop, and again, flame when I open. I just need a small amount, so I'm going to lightly touch my pre-inoculations. I'm going to flame and close. I'm going to take my slant, flame when I open, and then again, I'm going to take my inoculant, my inoculating loop, and I'm going to just rub it and drag it lightly across the surface of my slant. And then I'm going to flame, close, and then label. So now what we're going to be doing is we're going to be inoculating sterile media from a plate. So what we need to do here is, right now I have the plate closed. I don't need to flame it or anything, but I have it closed in my sterile flamed environment. And I'm going to lightly touch across this here, my plate. So I'm going to just lightly touch. So as you can see, I didn't drag across the plate at all. I just had a light touch against the surface where my growth is. Now I'm going to close the plate back up because it's no longer needed. And then I'm going to inoculate my broth by simply, in my loop, placing it into my sterile media with my pre-labeled tube. And now I'm going to flame it again. I'm going to close my tube. And then I'll incubate this at 37 degrees centigrade. So, what we're going to be covering now is streak to isolation technique. The reason we're on the board right now is because it's a little easier to see the principle of what we're doing on the board as opposed to simply just looking at the process when inoculating a plate. So, as you can see, I have my plate represented by this circle divided into 4 quadrants. So, I'm going to label these quadrants. Quadrant 1, Quadrant 2, Quadrant 3, and Quadrant 4. So, as always, we always have our loop that we're taking our inoculant with. It will always be sterile, so make sure that you flame your loop before picking your bacteria from whatever agar you're using, whatever inoculant, so your broth, your slant, your plate. And now what you're going to do is you're going to make your first pass of streaking. So I'm going to take my loop and I'm going to streak it across my plate. Okay? So between 1 and 2, I'm going to flame the loop. Okay? Once my loops cool down, what I'm going to do is I'm going to take my loop and I'm going to start it at the edge of number 1 that's been streaked. So I'm going to take this, I might go back one more time, and then after that, I'm going to spread across quadrant 2. And again, between 2 and 3, you are going to flame your loop. Now, once my loops cool down, I'm going to take my loop, and I'm going to start by dragging it through the end of 2. I hit it one more time, and then after that, I'm going to keep streaking. Okay? So, one thing to notice, when I'm doing this, I'm trying to keep my streaks a little spread out. So I don't want something, for example, like this. The reason being is since we're trying to isolate these colonies, we want to try to spread out their growth. Okay? So now, lastly, what I'm going to do is between 3 and 4, as always, I'm going to flame my loop. And now once my loop is cooled down, I'll take a small amount from the previous inoculation, streak it, go back one more time, and then make sure that you do not go back into your original streak. So this is kind of a drawn out theoretical practice of what we're going to be doing when we streak for isolation. So when you have your final plate, once everything is grown up, this is what you'll see. Green will represent our bacterial growth. So in 1, you might have a lawn. In 2, you're going to have something like a lawn with colonies throughout that are touching. So this is going to be more dispersed. 3, you might have growth that's almost isolated but not quite. So 3, you'll start seeing some isolation. And then 4, you'll either see little to no growth because you've isolated at 3, or you'll finally get isolated colonies. And then this is what we would do in our forward experiments with biochemical tests, or for any other kind of thing such as sequencing, or if we're going to transform. So the goal is to ultimately get to an isolated colony for any other future applications downstream. So now what we're going to be doing is a streak to isolation. So the purpose of this is to streak your bacteria into isolated colonies. So what we'll do is we'll start by taking some bacteria. Just a small amount. And what we'll do is we'll first start by streaking across the plate in a dense area. Okay? So this first area will be my area 1. Between each streak, I'm going to flame my loop. And one thing that's also helpful is to split your plate. So this plate that I have has 3 quadrants. So I did my large area, and then I'll be taking from the edge of my large area, which was over here, and I'm going to be streaking across here. And here, I'm always trying to leave as much room as I can between streaks. That way, I can decrease the density of my bacterial growth. So I'm going to close. I'm going to flame. Then I'm going to wait for my loop to cool down. So again, it's always important to label where you have your areas. So I can look at my plate and see where I've streaked to look at where my areas are. So I actually have a second quadrant now. So now I'm going to take the edge of my second streak, I'm going to drag it briefly in my second streak and maybe hit it again, and then after that I'll no longer streak from my second streaking. Now I'm going to flame my loop again. I'm going to wait. Another trick that you can use to make sure that your loop's not too hot is you find an area of uninoculated agar and you just tap. So if your loop is too hot, you'll be able to hear a small sizzle noise from the agar in contact with your warm loop. So now I'm going to do my last streak, which in this streak I am not expecting to have anything. So as I did my streak, I want to point this out. I streaked down, and then I streaked up. That is actually not proper. So the reason I showed this was just to show you what improper streaking might look like. So you'd never want to backtrack the direction of your streaking. So now, make sure to flame my loop, as always. And I'm going to make sure that my sections are properly labeled. So I have 1. So this was my first streak. I have 2, which is my second streak. 3, and then my last streak here. So I actually streaked in 4 quadrants. Now you should be able to actually streak for isolation with 2 different microbes in half a plate. So what that would look like would be taking this plate, I streak, streak. So right now we're not going to be worried about doing anything more advanced. We just want to make sure that we have the idea of streaking for isolation. So now what we're going to be doing is we're going to be making a pour plate. So to use a pour plate, we have our liquid agar. So what we're going to be doing is inoculating our liquid agar with our serratia here. So with my pre-flamed loop, I'm going to pick a small amount of serratia. And now what I'm going to do is inoculate my liquid agar. Okay. So I'm going to close this. Now, I'm going to mix up my Serratia by mixing up my liquid agar. And then I'm going to take my plate and I'm going to pour my liquid agar into my plate. Okay. And now what I'm going to do is I'm going to let this cool. And then I'm going to wait for my liquid agar to solidify. And this could be about 5 minutes or so before it solidifies. So you'll notice your liquid agar will have kind of a solid opaque color once it's solidified, but until then it'll look like a golden or a honey color.